Hey Molly.
Hello. Oh, I've still got my Pede sticker on.
Oh, nice.
How are you?
Yes, I'm good, thanks. How are you?
I'm good. Should probably move my plant as well. It's crowding everything.
That's
So was
fine.
there an electron before mine or something? I remember.
Yes, there was, but I've just restarted it actually because it finished a bit early.
Oh, cool. Good, good.
So hopefully we'll get some people joining soon. Uh,
Nice.
I think they've had an exam today or something like that. There has been loads of exams this week, I think.
Have they? Okay.
Or they've had, yeah, because I'm in their chat, I can see that they've had something on. So
Oh, you're the one like lurking around in the chat.
yeah.
They I remember when I actually attended this lecture, it was really unpopular.
Oh,
Like I feel like there was five people in it. I feel like people are going to avoid this question if it comes up.
well,
Um,
fingers crossed, got at least one.
Nice, nice. I don't know what the ideal number to have is. Is it better to have more? Because then they're actually someone might say something, but then they also get a bit scared if like to say anything. But I don't know. I'm just writing down the answers to my MCQ questions in case I forget them.
Yeah, so fair. All right, I will drop off now. But good
Okay.
luck.
Yeah, that's fine. No worries. So shall I just let them? Are you present in the background anyway? Or
Yeah, yeah. I'll just like put my camera off and everything and
yeah, yeah, fine. I'll just let them all join and then do it. Is it an hour, or would you like to do it in a bit less? Or
completely up to you.
okay, I'll make it less than an hour so that they can like
Nice. All right.
stay earlier. But cool. I'll maybe start at like 3 or 4 minutes past so that people have time to join. Okay. Thanks guys for joining so far. I'm just gonna wait till like 3 minutes past maybe in case other people join. Um, and then I'll start sharing slides and everything. So I'll just give it a couple more minutes. I think I'm going to start now. So I'll just introduce myself to everyone. And there's not many people here because I think you've had an exam today, but this will get recorded anyway, so you can all watch it on there anyway. I'm going to share my screen and do and the slides will be available as well. And just a note on the slides that they do have kind of notes in the annotations on the bottom of the slides. So things like when we do MCQs or when there's some other like diagrams, there are kind of additional explanations in the notes as well. So don't stress too much about making loads of notes and things like that. And if you have any questions throughout, just unmute, just in case I don't see you raise your hand or anything like that, but literally just shout out and stop at any point. And that's fine as well. And then, yeah, hopefully you should get recorded and you should get the slides at the end if you fill out the feedback form. So if you could do that, that'd be great as well. And yeah, so hopefully this won't take a full hour, or I'm not sure how long all the rest of your revision lectures have been. But yeah, so feel free to stop me at any time or ask any more questions. I'll try and cover things that are on the spec, and I won't go too far outside of that as well. So I want to keep it more relevant as well. So hopefully that's useful. I'll start sharing my screen. Could one of you be so kind as to let me know if you can actually see the slides and hear me okay, just so I know? Yeah, someone's given me a thumbs up as well. I might, there is a bit of like optional participation. There are some MCQs to go through. I understand if some of you aren't in a space where you can respond, but also just it's nice to give my voice a break if you guys want to actually answer some of the questions as well. And they'll be kind of like mock like your MCQ questions might be. So feel free to have a go at some of those. That would be great as well if you could. And yeah, ask any silly questions. I've probably heard it before. So this is what we're covering today. So if I go onto the spec and show you what I'm trying to cover, we'll do it in a bit of a reverse order. So this comes later in the spec, but first I'm going to quickly go over necrosis, apoptosis, pyroptosis. They're just things that you need to know about. Less likely that you might get like a full essay question or something like that on this in itself, but it's just useful to know and to know the basic mechanisms of each and when they're seen. We'll then go over the key mediators and cell types in acute inflammation. We'll go over kind of the leukocyte rolling and everything that happens in the kind of post-capillary venule and in the endothelium. We'll go over some small bits of clinical relevance that you could put in your work as well that is quite interesting in relation to the physiology. And this won't cover anything kind of specific immune response related. So like your B and T cells, that's in a separate lecture that you might have already had. But this will just be more the innate side of things. And then from that, we'll kind of focus on the function of neutrophils, macrophages, and then some of the specific ways that they kill pathogens as well. So hopefully we'll cover at least briefly many of these things. If I don't talk about it in detail, it will be on the diagrams. It will be in the slide notes as well, hopefully. And obviously I can't cover absolutely everything because it is a huge chunk of the syllabus to be honest to do in less than an hour, but I'll cover some of these things and hopefully I'll point out if there's things that could be useful for essays and things like that. So I'll get started. So first of all, I was going to talk about apoptosis and necrosis. So generally it's referred to as once there's irreversible cell damage, the cell needs to kind of, not that it's got a conscience, but kind of decide how it's then going to die. And will it do that by in a programmed way that's organized and anti-inflammatory, or will it do that in kind of a pro-inflammatory and disorganized way? So it can do that through apoptosis, which will be controlled. It will form these apoptotic bodies. They're taken up via phagocytosis, and those will be cleared by phagocytic cells. These include cells such as macrophages. You've probably had lectures by either Professor Greaves or someone similar about macrophages. As you know, they come in different types, and some of them have different subtypes, M0, M1, M2. And some of those subtypes are associated with being either pro-inflammatory or anti-inflammatory, and those are kind of associated with the apoptotic mechanism of maintaining homeostasis and being more like tissue resident as well. And then on the other hand, you have necrosis, which is more of an uncontrolled cell death, and that will be pro-inflammatory. So that can, in a way, that kind of heightens the inflammatory response, but it would also kind of cause this cell kind of releasing of its contents, or kind of think of it as kind of like exploding cell in a way. The diagram at the top kind of illustrates it quite nicely as well. And it's very, think of it more as a very uncontrolled mechanism and pro-inflammatory. Whereas if you think of apoptosis, remember the forming of apoptotic bodies, the shrinking of the cells and the shrinking of the DNA, the chromatin condensing. Remember, it forms the membrane buds, and then it's also anti-inflammatory, and you're forming those apoptotic bodies and resulting in kind of caspase enzyme. And those will be the enzymes that are kind of controlling the apoptotic process. Necrosis being inflammatory, so that will release these things called DAMPs, which are damage-associated molecular patterns, and those will cause inflammatory cytokine release. So those cytokines might be IL-1, you'll hear of quite a lot, also IL-6 and IL-12. Whereas apoptosis will be more of resolving inflammation rather than promoting it, and that might have associated cytokines such as IL-10, for example. Another one to note is pyroptosis. So that will be the death of kind of bacterial infected cells. And when that cell contains the bacteria, it will then die, and in doing so will kill the bacteria within it as well. And there's some diagrams here that kind of explain it here. And especially here, you might want to learn about the Gasdermin protein and its role. And when it is in the mechanism of killing the cell, it will be inserted into the membrane, and that pore will really cause kind of like the lysis and the death of the cell. Another thing to know about is the caspase enzymes. So they're quite important to know about. I'll just show you this diagram, but not that you need to worry about it, because I think this is more kind of extension stuff that is not core knowledge. But just to know that there's two different kind of main roles. There's an intrinsic and an extrinsic pathway. And this will involve the role of caspase enzymes, like I mentioned before. Especially caspase 3 and 7 are important to remember in kind of having that active role in cell death and apoptosis. So the intrinsic pathway, which is on the right-hand side, will involve cytochrome C and activation of initiator caspases, and they kind of trigger other caspases, which are called executioners. So you have your initiator caspases that start, and then your executioners here. Executioners will be 3, 6, and 7. So 3 and 7 is in this diagram here. And then you've got the extrinsic pathway, which might happen through this receptor, FAS-L. And it can also happen through the TNF alpha receptors as well, with their appropriate ligands, TNF and FAS ligand binding. And these will also then merge on the caspase 3 enzyme initiation, which will trigger the initiation of apoptosis as well. You might remember from when you do your cell membrane biochemistry as well, that you have kind of the phosphatidylserine, and that's important because it's usually on the inner leaflet of the membrane. And when that's flipped onto the outside as well, that's an important process that happens in apoptosis too. So if you want to know kind of the more intrinsic things or the ins and outs of it, and also there's a lot of research into caspases and how they're useful, because if you can think of how we could pharmacologically manipulate a caspase enzyme or use that, or understand more about how it's involved in certain pathologies or cancer cells, for example, that's something you could talk about as well, which is quite interesting too. But not essential, so don't worry too much about this, but just that there are these different things involved. And BCL2 as well is an important gene that you could also look at too. So I don't know if you want to have a go at this question. So which of the following statements is most accurate in relation to necrosis, based on what we just kind of spoke about? Does anyone want to have a go at guessing? So I might have to pick on someone if anyone wants to have a go. Seon? Am I pronouncing your name correctly? Or Beverly? Whichever names are first. If no one's in a... Okay, there's someone in the chat, sorry. I didn't see that. Someone said E and someone said, yep, B. Okay, so E is the correct answer. So the reason it's E is because of the necrosis is the pro-inflammatory response, like I said, and release of the cellular contents and kind of the pH changes as well. You do get like lysosomal leakage. If you remember, the role of the lysosome in the cell is with that lower pH. So you do see E. The rest, A, B, C, and D, are things that you do see in cell death, but you see them in apoptosis rather than in necrosis. So this refers to like the apoptotic bodies that are formed, the membrane blebs, sorry, and then they go on to form the apoptotic bodies. Chromatin condensation, that was very organized, that's seen in apoptosis. Eosinophils ingesting the cellular contents, that's in apoptosis too. But yeah, so E is on that you see in necrosis. I didn't specifically say it, but if you could logically guess that that might cause a more pro-inflammatory response. But yeah, sorry for picking on you. I didn't realize you in the chat. Oh, sorry, let me stop sharing and reshare my screen. One sec. And we'll carry on. Okay. Okay, looking at that. Can you see that now? Hopefully that's fine. So I'm going to generally talk about innate immunity and the role that that has as well. Thanks for the thumbs up. So generally, innate immunity in the innate immune response, we're talking about it being fast. So it says within 4 hours, there'll be an established response, but it essentially is kind of minutes to hours as well, and can last up to days. If it's going longer than the kind of several days process, we would either call that a repeated bout of acute inflammation, or that might start to turn into chronic, which we'll also touch on. It's non-specific, like I said, it's not with those B and T cells, so it's not necessarily specifically antigen dependent with those kind of formulation of antibodies, but it's more to do with the innate response that the body produces from different cell types, regardless of the specific type of pathogen itself. It has no memory as well, so that's important to remember. So it's not like seeing the same pathogen again. You would have to mount exactly the same anti-inflammatory response, even if you then might mount a more specific kind of memory response from your B and T cells. So there's the cellular response immediately. We'll talk about the different cell types that are involved. The chemical response through cytokines and complement. So there's a lot of secreted mediators, and these are kind of also termed soluble mediators, and there could be lipid-derived mediators we'll talk about. These are also the basis of a lot of our anti-inflammatory drugs as well, like kind of steroids and NSAIDs that we'll talk about too. And then this initiates the inflammatory response. So this is quite a nice phrase. I know it's in Latin, it's a bit silly, but it kind of means heat, redness, so erythema, tumor will be the kind of swelling, I think, or edema. Dolor would be pain, and then this one means kind of just loss of function of whatever has undergone the inflammatory process. So this is quite a nice summary that you could kind of include in your explanation of the innate immune response. So if we consider the inflammatory cascade in general, we're thinking about, say, if there is a site of injury or pathogen, so you've maybe got an infected cut or wound in the skin, just imagining that as our kind of example here, and the surrounding capillaries and post-capillary venules, which is where we'll be having kind of the immune response will be leaking into the tissues in order to help fight off that infection at the site or at the source. So the first thing that's important is sensing that there's something that has gone wrong, whether that be through damage to the tissue or through a direct pathogen present within that area of tissue. So the damage would kind of release damage-associated molecular patterns. So is a cell injured? Has it burst? Has there been a wound? You're going to be releasing cellular contents. These aren't supposed to be just really happily floating around. So this is the kind of thing that would trigger an inflammatory response because it's somewhere where it shouldn't be. And that's because it's been detected by these tissue resident cells. And like I said, the types are kind of macrophages that can change their subtype based on what's going on. These macrophages might well be just kind of happy and anti-inflammatory until they notice that something like a pathogen or an injury has happened to the cell, in which case they're going to be sensing these DAMPs or PAMPs, which are associated with pathogens, and then they're going to be initiating their responses. So just imagine these little blue spots of bacteria as well, and these are going to be recognized by our tissue resident cells. They could be dendritic cells, quite often in tissues, or they could be Kupffer cells. So those are found in the liver, I think, and microglia found in the brain. So these tissue resident cells are really important for antigen sensing and that initial inflammatory response, and whether to kind of inflate it and whether to intensify it more or whether to dampen it down as well. So the PAMPs that we always consider is LPS, lipopolysaccharide, which is associated with certain types of bacteria. Exotoxins also released by bacteria can be picked up as well, and they're very pro-inflammatory in themselves. It's just generally important to know that these are picked up by specific receptors. You don't need to know loads about these receptors, but if you do, it can kind of like enhance some of your responses as well. So there's different types. There's toll-like receptors, which are the TLRs, NLRs, nucleic acid receptors, kind of detecting different types of a DNA or RNA. So if it's specific types of strands or single double-stranded RNA, could be the genetic material of the bacteria, for example, that is being picked up as well. It's important to note that there's also the initial barrier response. So the skin itself, mucosa itself is very protective, and that's got high quantities of immune cells in it naturally. You also got things like gastric acid, bisalts, and the microbiota of the gut itself, which is generally forms a really important barrier function. But obviously that barrier might break down. So that's why it's important to have these kind of tissue resident cells, which are there to kind of initially sense those pathogens. So sensing via specific receptors, generally called pattern recognition receptors, PRRs, and these are these specific types of TLRs, NLRs, and these are all present, different varying types, but they'll be generally present within these tissue resident cells and other antigen presenting cells, which are kind of floating within the tissues themselves. So this is all within the tissue. We're not yet kind of looking at the endothelium or the capillaries or venules yet. So the next slide I'm going to briefly skip over, but it will be in the slides to you to look through. But these are just going into detail in the different types of receptors. Say if you wanted to talk about a specific receptor, there's lots of different experiments which have been done on these types of receptors in figuring out where they're relevant, if they're overactive in certain autoimmune conditions or different disease pathways. One of these, for example, is TLR4, is really important in sensing lipopolysaccharide, which is kind of a really classical example of an innate immune response, an innate receptor immune response. There's probably lots of research on this specific receptor interaction. Basically, the main point of most receptors being, as in lots of other subjects you've been taught about, is triggering of an intracellular cascade, and that will release that will activate different kinases and then result in differential gene transcription, and that will affect the behavior of all the cells in that area. So this is generally going to be pro-inflammatory and kind of promote that pro-inflammatory response. It's also relevant in septic shock, but I'll let you read through these notes yourself because it's not particularly essential for you to read through. There's also such thing called the inflammasome as well, which you can also look up. I don't want to go into it in too much detail, and there's some notes on the slides on it too. But there's different types of inflammasomes. They've got different names and essentially result in the different, they can help promote different types of cell death and also death of the pathogen itself, and helps kind of boost up that pro-inflammatory response too. Cool. So the first stage is detection, and we've detected and recognized that there's antigens present, there's bacteria present, they've been releasing DAMPs, PAMPs, and they've been recognized by our antigen presenting cell. And by our initial tissue resident cell, whatever cell that might be. So this needs to start off the pro-inflammatory response, and it will do that by releasing certain cytokines and certain mediators. And then it will also, other than outside of cytokines, there's also those kind of pro-inflammatory lipid mediators that you might have heard about as well. So things like prostaglandins, prostacyclins, and those will act where NSAID drugs act as well. So all of these different mediators will be released. What also will happen, as we can see here, you've got your normal red blood cells and your kind of polymorph cells here. You see they're polymorph because they've got this interesting shaped nucleus. You can see it's slightly segmented. And you've also got your other types of white blood cells in the blood here. What will happen where you've got these pro-inflammatory cytokines released, you'll get activation of these endothelial cells, and they will help kind of push and guide these polymorph or neutrophil cells towards the site of tissue injury or towards the site of where the pathogen's present. So your red blood cells, in addition to the kind of lower flow state, will start to form what's called ROLU. I'm not very good at pronouncing it, so that might be completely wrong. But these will help kind of clump up as well. And these red blood cells are clumping within the venule, and that will help to kind of slow the flow down to make it easier for these cells to move through the endothelium where they can be active and where they can help kill the pathogen. So the next step is leukocyte rolling. So it's kind of adhered to the endothelium, and now it knows that it needs to kind of start to move along the endothelium to get to the specific site of injury. So it will do that through rolling, and the rolling is mediated by selectins. So as you know, it's not supposed to be a very fixed interaction, so it needs to be weak. So just to learn that selectins are weak, and then the firmer adhesion, when they need to stop rolling and move through the tissue, that's mediated by integrins and ICAMs, and that will be a firmer interaction, whereas the selectins are the weaker interaction as well. These are all attracted again by positively chemotactic gradients. So the cytokines and all the inflammatory mediators released at the site are going to be attracting that neutrophil to where it needs to go. Once it's stopped rolling, it will then need to move through the endothelium. It does this through a process called extravasation or diapedesis as well. So just to know what those terms mean. And it will move through the tissue in that way as well. Once it's moved into the tissue, it's then kind of at the site where it can kind of activate itself. There's no point it being active in the venule where it's not going to have an effect. It needs to kind of get through into the tissue first. Similarly, you've got your tissue resident macrophages, but a really important source of macrophages also comes through the blood itself, derived from monocyte cells, and those are derived from the bone marrow. So those will go through the blood like the neutrophils do, and they will enter the sites of tissue injury. They're not originally present there necessarily. So you also get influx of further macrophages, and they'll be derived from monocyte cells. So it's good to know that there's two different kind of origins of those macrophages as well. The positive chemotaxis as well, I just want to mention is, like I said, it's through chemokines, and those are acting through G protein coupled receptors. And then once they've reached the site of injury, they'll then be presented to the pathogen via these kind of presenting cells. Cool. So that's most of that stage. So yeah, as we said here, once it's entered the site, we'll go through now the different mechanisms in which those innate immune cells will then kill the pathogens. And sorry, that's what I explained there. I didn't realize it was on the next slide. So there's different mechanisms. There's non-oxidative and there's oxidative mechanisms. There's phagocytosis, like we said as well. And then the neutrophils themselves have slightly more specific ways of also killing pathogens as well. Cool. I think that's all I wanted to go over. Anyone have any questions from that so far, or anything they wanted to ask? Nothing so far, so that's probably fine. Cool. So thing I just wanted to mention before we go into the specific cells and what they do is, in general, there's this term called the acute phase reaction or acute phase response. So you've probably had this in a Professor Greaves lecture maybe, but it's essentially talking about some key cytokines that are released and some key actions which happen in the initial inflammatory response. And there's a big involvement with the liver as well, and the types of proteins that the liver then starts to produce. And that's all just part of this acute phase reaction. And it's quite important clinically because it might change some of the blood test results that you see as well. And it could be that something is raised or decreased in the blood results, and you're thinking, why is that? Could it be due to an inflammatory response, for example? Two important interleukins or cytokines to know about is TNF alpha. You'll hear about it loads, but if you had to learn two, I would probably learn these two. And interleukin 1, so they're causing systemic effects themselves. So IL-1 will act to kind of raise your temperature set point. I think it acts in the hypothalamus and centrally to help kind of raise that set point so that you might spike fevers. And that's essentially kind of beneficial for you trying to kill a pathogen or rid the pathogen from your body. And then you've also got TNF alpha, which is very pro-inflammatory and is associated with lots of different diseases as well. But this in itself will then have ongoing effects acting through TNF receptors, and that will upregulate many other inflammatory proteins, complement proteins. And in doing so, it's kind of shifting the types of proteins which are made. So it will make more inflammatory proteins and it will decrease other proteins that it doesn't want to make, such as albumin, for example. it's doing this through kind of conserving amino acids so that they're useful in the inflammatory proteins and not being used on other things. Other things which go up, you might see ferritin going up in the acute phase response as well. That's something that can be measured as well. Cool. And then that's just acute phase, just to mention. And then essentially we're going to talk about now these different things which happen during the innate immune response and those effector mechanisms. If it's not particularly detailed, because I want to get through as many slides as possible, then the notes will be in the slides as well, if I don't have time to go through every single one in lots of detail. Just a note on some like clinical relevance of why this is all important. So for an initial inflammatory immune response, the acute immune response is very amenable to certain different drugs. These can include NSAIDs, which are non-steroidal anti-inflammatory drugs. So steroids are different types of drugs that we can use, but what you might use in the short term might be something that's non-steroidal. These include ibuprofen, aspirin is a type of NSAID, technically paracetamol is as well, although it acts in a slightly different way. Ibuprofen, naproxen, I think I already said that one. And then there's different generations of NSAIDs as well. There's first generation, which inhibits COX-1 and COX-2, and there's also second generation, which is more COX-2 specific. And they're called the COXIBs. So basically what they're doing is, like I said before, all the inflammatory mediators which are increased and released by those tissue resident cells, they include these different types of lipid mediators. So it's not just the soluble cytokines that we spoke about, it's also these kind of lipid mediators as well, like prostaglandins, prostacyclins, and this other one called thromboxane A2. So what these do is also help kind of further increase the inflammatory response in the inflammatory cascade, and they're kind of automatically increased and ramped up when there is inflammation. So what we can do is we can inhibit these. So NSAIDs will be enzyme inhibitors, so they'll be cyclooxygenase or COX enzyme inhibitors. So classical one of this, an example would be aspirin. And aspirin, as you know, it can be used as a pain reliever. It can also be used as an antiplatelet drug, and that's because like the thromboxanes have roles in platelet aggregation and things like that. So aspirin's a good example of that too. Ibuprofen, like we said as well, and that's how it increases its kind of like pain relieving properties as well. And just know that comes from the main mediator it comes from is arachidonic acid as well. There are other pathways and they're called eicosanoids, which are released as well, which are those lipid mediators, but these are the main ones to know about. I think there might be a couple others which come up too, but yeah, that's okay to know for now. These are some of the side effects. You can read about them in your own time as well. Just to know that all drugs come with side effects, so it's important to think about those too. And then that's contrasting with the mechanism of action of steroids, which will be more through a nuclear mechanism. So when you think about how drugs work, if something's acting through an enzyme, it's going to be acting quite quickly. If something's acting through nuclear receptors and through transcription and translation, as steroids do, it's going to be acting a bit more slowly, but then its effects will also last longer. So that's why steroids are a good example of more of a long-term immune and associated with more kind of immune suppression, but it will also dampen down that inflammation in a longer-term period as well. Something interesting to look up with, I think there was an interesting trial done looking at the use of steroids in severe COVID as well at the time. I think that was done in Oxford, where they looked at the use of dexamethasone, which is quite a cheap steroid drug, really, and it worked really well in patients with severe COVID to help their dysregulated immune response. So there's lots of examples where steroids are useful whenever there's a component of an overactive immune response or a pathological immune response. You might find that steroids can be used later in management, although they will also, it is that fine balance between immune suppression and kind of treating the dysregulated immune response. So just an introduction of those two drugs, and you can kind of talk about those if you're interested. So one of the main ways in which these kind of initial innate immune cells will be killing pathogens will be through phagocytosis. There's lots of different kind of really complicated diagrams and receptors involved and enzymes you can learn, things like that. But just to go over the basics is that there's various cells which can achieve phagocytosis. So it's not just macrophages, it's neutrophils. Natural killer cells are also able to do this too. And dendritic cells are a type of tissue resident cells which do this a lot. It's important for antigen presentation as well. And then it's also important for sending off these kind of eat me signals and helping promote the pro-inflammatory immune response too. There are various enzymes which are really important. So just to know that but when we're looking at oxygen-dependent antibacterial killing within a phagocytic cell, there are different enzymes which are important in different cells. So INOS, which is inducible nitric oxide synthase, is really important in macrophages. And then NADPH is really important in neutrophils. And this is an oxidase enzyme. Sorry, there should probably be an oxidase there. And that's really important in neutrophils. And we'll talk a bit more about what might happen if you don't have that enzyme later as well. So there's the oxygen-dependent mechanisms where these enzymes are really important in creating radicals, and these radicals will kind of form into other different byproducts, and these are very toxic to bacteria, and that's how it will kill them. And then there's also the oxygen-independent mechanisms. So, for example, the neutrophils will have things called neutrophil extracellular traps as well, and they will also release lots of different pro-inflammatory and kind of degrading enzymes as well. So that's also how they will achieve killing too. And there's also kind of the killing of bacteria itself within the lysosome as well, obviously. We'll talk about nets a bit more when we go on to neutrophils. And there's also these reactive nitrogen species, which are created by the INOS enzyme as well. And just a note that there are some pathogens which are famously very good at escaping phagocytosis and are kind of escaping the phagolysosome. There's quite a few which are very good at this. And one that you could think of might be tuberculosis as well, where they really struggle to kind of kill the initial bug through this initial mechanism. So there's other mechanisms which have developed, which I'll also talk about as well. Cool. So I think that's the main things to talk about with that one. Talking more specifically as well about the respiratory burst. So this is when we're thinking about the different radicals which are made. This definitely requires the use of oxygen. So there's various experiments which are quite nice to talk about in relation to this, where they've kind of looked at oxygen consumption in all of these cells over time. And as you can see that as they're active and as there's pathogen present, that they are degrading through this mechanism, you can see kind of the oxygen consumption kind of increase within these cells because they're using the oxygen. And they're using it through these different oxidase enzymes to make these free radicals. They make very bactericidal hydroxide radicals, and then those are really useful in killing the bacteria. Clinically, it's quite important they're very rare, but there are some disorders where there might be defective enzymes, so defective NADPH oxidase. So you might see that in this disease called chronic granulomatous disease. And I'll let you look that up in your own time as well, but essentially it's X-linked, so it tends to affect primarily boys. And you might see, well, I would ask you guys what you might see. Oh, someone's put something in the chat, sorry, let me have a look. How does phagocytosis in neutrophils and macrophages differ? So essentially, if I go back, so it differs via this mechanism with the different oxidizing enzymes, but then it's also generally the time at which they're undergoing the phagocytosis as well. So they will both be performing phagocytosis, but it might be that they're doing that initially as a tissue resident macrophage, and they might be using this inducible INOS enzyme, whereas NADPH oxidase is a really important enzyme used in neutrophils, which you won't find as the mechanism in macrophages. I can go back and respond to that question a bit more later as well, if you like. And yeah, so essentially, if you had a problem with the NADPH oxidase, there's a problem with the neutrophils, and you might need more neutrophils to be mounting that response, a similar response to someone who's got a defective enzyme. You can diagnose this through this assay as well, which is also interesting to look at as well. And I won't go into too much detail because I'll run out of time, but essentially in patients who are affected with CGD, you'll see a different, the NOI index, the oxidative index, you want it to be above 100. So their ability to kind of oxidize and their ability to form this anti-inflammatory response and the creation of these radicals, they should be able to do that. And there's an explanation of how this kind of experiment works in the comments as well. But you can see that there's a different shift and a massive drop in the NOI compared to healthy controls in people who are affected with the disease as well. And just to note that the DHR and the PMA is what we're adding to replicate that kind of stimulation of the oxidative burst. So those are kind of the mechanical things that we're adding. And then if you look here, this is kind of the control experiment, just to show that it's not the addition of the DHR, it's the addition of the PMA that is making us stimulate the macrophage. And that's when we see a big difference in the oxidative response. And in those who were affected with the disease, we see a big reduction in the oxidative response. Something I mentioned earlier was the neutrophil extracellular traps as well. So neutrophils will extrude their cellular content through their own death. So say they know that they've engulfed some bacteria, they've engulfed a pathogen, and that cell needs to die. It's kind of it's a very pro-inflammatory form of cell death as well. And through this, but when it dies, it will form this trap, and that will contain lots of histones and granule proteins and other kind of proteins and bits of enzymes that, as I said, they're not supposed to be found in that extracellular space, so they'll be very pro-inflammatory. But what they do is they're all kind of webbed together, and they will literally form kind of a physical net or trap that in the death of the neutrophil will still be kind of performing a useful function in trying to catch more pathogen. So that's why it's quite an important mechanism too. There's also been lots of research in how it can be pathological as well. So how maybe there's kind of overactivation or dysregulated net formation in certain diseases. So that's quite interesting to look up too if you're interested. Cool. So just remember that respiratory burst and NADPH oxidase is very important, and you're killing it through the use of oxygen, creating these really bactericidal free radicals as well. Cool. So this is a bit more clinical, and I'll just skip through it quite quickly. But just to know that if you've got increased neutrophils, say you just had a blood test and there's increased neutrophils that you're seeing, why might that be? Well, as we know that they're really closely associated with the bacterial infections, and they massively increase when there is a bacterial infection. So think about your common causes of bacterial infections, such as Staph aureus and Streptococcus. Those are called pyogenic bacteria, and pyogenic meaning that it will be pus forming, and kind of sometimes associated with, like generally feeling systemically unwell, fever, things like that. And that might cause your neutrophils to go up. That's the really important one to remember. The other things are a bit more kind of small print, but it's there in case you want to know it. And then decreased neutrophils, just to show you, because I know you still do quite a lot of histology as well. Say your bone marrow is failing, and as we know from that big family tree of all the different white blood cells, the bone marrow is the starting point of generation of all of these different cell types, from the hematopoietic stem cell. If there's something wrong with your bone marrow, you can't then generate your neutrophils. So this here on the right at the top is the bone marrow, which you can see has been replaced with loads of fat cells. That's because there's aplastic anemia, and it's kind of all formed a big fibrotic stroma, because there's no more hematopoietic stem cell generating new blood cells. That can be caused by lots of different things, and also by drugs. And then at the bottom you can see here there just seems to be a lot of elevated destruction of neutrophils as well, and you can see that in certain genetic diseases as well. And yes, you can look up some of these really rare and wonderful diseases as well, but they're very, very rare generally, but not associated with very, very good outcomes, unfortunately. Okay, briefly on degranulation. So other different types of innate immune cells are mast cells, basophils, eosinophils, less prevalent cell type in the blood, but they're still really relevant, and especially relevant in this allergic response as well. And this response can lead to anaphylaxis and anaphylactic shock. So the exact kind of steps of it I won't go through too much, but essentially you're exposed to an allergen, and that allergen is not necessarily a pathogen. It could be a pollen, a tree pollen, but that's going to bind your B cell receptors and that will be specific to a B cell receptor and cause a specific IgE antibody to be mounted onto those FC receptors, and those are present on your mast cells. So these mast cells here will have a specific IgE for that FC receptor. That's called the sensitization process. And basically the next time that specific allergen is kind of exposed to the individual, whether that's because they've digested something or it's like an antigen present on like a nut, for example, that specific allergen will that specific antigen will be present. That will trigger the same IgE to be triggered. And that will cause the mast cells to be activated. Those IgEs will cross-link, is quite an important phrase to use. And then those mast cells, once activated, will then go on to degranulate. So they will release all of their granules outside of the cell into the capillaries, and those will spread around in the blood. Essentially, this is known as kind of a primary mediator cell type, and that's because it contains all of its kind of active mediators through the granules as well. Cool. And then we'll act on H1 receptors having differing roles, but those H1 receptors are really key in causing the smooth muscle contraction, which will cause your bronchoconstriction, vasodilation, which will cause your low blood pressure. And then it has various other cell effects on different parts of the body, like cardiac stimulation and gastric secretion, things like that. You might have been spoken about the wheel and flare reaction before. So it's the same thing. If you kind of, I think it's like the classic example is like rubbing yourself with a ruler or something like that, where it goes red, you can kind of cause the same degranulation of mast cells in the tissues, where it kind of has that kind of red response and then goes white afterwards. That's where you've seen that degranulation happen. So obviously they can be useful in a non-pathological sense, but this is also very relevant in the treatment of anaphylaxis as well. And we'd reverse that vasoconstriction with adrenaline. We use antihistamines and steroids a bit less nowadays, but adrenaline is still the main point of treatment as well. Okay, so the next point to say is opsonins and complements. So we're talking a bit more now, less about the cell types, but more about the mediators which are released as well. So we talked a bit about the arachidonic acid derived mediators, but these are also important kind of amino acid based mediators and complement proteins. So these are really important as well in opsonization, which you could think of as kind of decorating your pathogen so that it's more digestible, so that your phagocytes are more likely to be attracted to it and to digest it as well. It also helps with antibody dependent cytotoxicity as well, which is important. So helps decorate it and attract it to both those innate immune response cells, but also to kind of the adaptive immune response as well. Generally just helping other cells, which is why they're kind of called complement, because they're complementary. And just to know that you're born with this, it's innate. You don't develop them over time as well. So just to remember that too. These are the specific receptors that you can learn, and just know that complement 2 deficiency is the most common complement deficiency. So if you want to talk about one, that one might be the most relevant. So you might see some more susceptibility to certain infections with that as well. Some pathogens are really dependent on complement and it's really important, and in others it's less important as well. So that's something interesting to know as well. Okay, this is the complement pathway. I'll leave it here. I won't go into it in loads and loads of detail, but it's generally important just to know that there's 3 different pathways. And essentially they merge on the C3 convertase pathway. And other important things to mention is the C5 convertase. C5 convertase, I know it's lots of horrible numbers and letters, and there's no way that you would ever need to draw this out, but maybe just to know specific parts of the pathway is important and how they're relevant clinically as well. So there's something called C1q deficiency as well. So you could think about that might be an important one. C3 convertase is really important and really helpful with opsonization as well. And then C5 convertase is really important in creating this MAC, the membrane attack complex. And that's really important for forming pores within bacterial cells, and kind of lysing those cells open as well. So that's why that one's really important. So that's C5b, C9 as well. But just remember, the C3b itself is really important for opsonization. C3a and C5a are really important for phagocyte recruitment. And they're also helpful in promoting mast cell degranulation and things like that. And then just to remember, this MAC complex is really important for lysing cells, forming holes in them. And that's really important for killing specifically gram-negative bacteria. Okay, that was just another question as well. Okay, so what I'll answer that question on IgEs when we get to the next question, if that's okay. Just the next MCQ, just in case you run out of time. But there's a question on mast cell versus eosinophils, but we'll answer that when we get to the next question. Oh. Okay, so other secreted molecules that are generally important to know about is the kinin pathway as well that you can look up, bradykinin, platelet activating factor. Those are all really important as well. They're not very large print on your spec, so don't worry about them if you're not interested in them, essentially. This is a list of all the kind of secreted kind of the formed lipids and lipid mediators as well. So just important that you've seen them and that you're aware of them, and they might be involved as well. Bradykinin's important in local sensitivity as well to pain, so to stop you kind of injuring the same area that you may have already injured previously. And then also interferons is essentially a really important part, and especially important with viral infections in the innate immune response to viral infection as well. So that's important to think about. Okay, but yeah, I wouldn't worry too much about this pathway, just to know that they exist, but you don't need to learn this at all. This is just for reference. Okay, so macrophages themselves are just, it's really important to know like a few facts about each cell type and what makes them distinguishable. So the macrophages themselves, just to know some of the basic markers could be important. So CD14 positive will be for the monocyte form, and they have a shorter half-life. But the tissue resident macrophages we were talking about will be CD68 positive with a longer half-life. And those will have all your differing phenotypes as well. And then just to know it's important to know the two different origins is quite a common MCQ question. So your macrophages might come derived from monocytes in your bone marrow, and they can change into your M0, M1, M2 subtypes. Or they might come from the embryonic yolk sac derived macrophages. They'll be self-renewing, they're your tissue residents, and they also have specific names based on what tissue that they're found in. Notably, osteoclasts themselves are derived from macrophages as well, so they can have really differing roles, and it's less kind of immune, it's less so much of the really important immune role, but it could be more to do with kind of a homeostatic role, like, the osteoclast might do in bone remodeling, and things like that. You need to know briefly about like the different types of macrophages. So you can have macrophages in different states, and depending on the different cytokines and the different kind of environment in which they're in, they will be more likely to be a different type of macrophage. So an M1, M2, or a regulatory macrophage. So just to know that the activated macrophages, they can be pro-inflammatory, or they could be less inflammatory or regulatory macrophages. IL-10 tends to be an anti-inflammatory cytokine, so just remember that one as kind of more anti-inflammatory as well. And that could be more kind of pro-resolutionary rather than kind of helping kind of create or intensify that inflammatory response. Okay. And just a side note that tumors as well can help influence the different types of macrophages in the area, so cancerous cells as well. Okay, so there's a question here about cytokines. So if anyone wants to have a go at this one and comment as well in the chats. There aren't too many slides left after this one as well. Oh yeah, and your question. So what is the difference between IgE mediated mast cell? Okay, so I'd say the mast cells are more associated with, I guess we tend to mostly talk about them in terms of pathology when we're talking about things like anaphylaxis as well, but obviously they're really important generally within like certain parasitic infections and things like that as well. So if you look at, I think generally, I don't know the exact answer to that question, so I'd have to look it up for you as well. But generally we think of the mast cell response in terms of the anaphylactic response and the massive degranulations as well. But you also see degranulations with eosinophils as well. I'm not sure which one is more predominant actually. I think you sometimes get a breakdown of the different cell types in the blood as well, don't you? I don't know if you have to learn those two. I'm not sure if you do. But I'm not sure if there are more eosinophils in the blood generally. I'd have to look that one up, sorry. But they're both capable of that degranulation response, which is the key thing as well. For C1q, it seems to come up a lot on MCQs. Do we just need to know they are associated with classical activation? Or I'm not really up to date with your spec, but I would imagine I would learn. So I'd say I would learn that they're associated with the classical pathway, and to maybe have a vague idea of which complement proteins are associated with which pathway. Basically, it's easier to learn the differences between the pathways, or the important differences between them, and then learn where they converge, rather than worry too much about learning every single step of each. And then obviously, if you do lots of questions, you might see which ones come up as a bit more relevant in your exam questions, and one which might be a bit more relevant clinically as well. Those might be more likely to come up rather than the really, like, the list of every single one. And definitely learning the convertases and the ones where they kind of merge would be quite important. Okay, no one's wanted to answer the question, but that's okay. So for this one, which of the following is incorrect about cytokines? So we know that they're kind of released mediators. So which any ideas at all? They do mediate natural immunity. Oh wait, natural immunity. No, they don't. Sorry, it's which one is not correct. So they're not going to have a role in the innate immune. They are going to have a role in the innate immune response. They're not going to have a role on lymphocyte growth differentiation. Nope. They can activate inflammatory cells. Yes, they will be. They themselves are not able to opsonize pathogens. So it would be D would be the answer here. So when we looked at the different things which can opsonize pathogens, it will be those proteins and complement proteins. So it wouldn't be the cytokine, because that's a released kind of soluble mediator. So that will act on specific cells through cytokine receptors. It won't be able to opsonize a pathogen in itself. Cool. For this one as well, we'll skip through it really quickly. Which is incorrect in relation to innate to acute inflammation? So with this one, I'll give the answer, but this one would be someone's put A. Unfortunately, it wouldn't be A. There is some contraction. You know, I said before there's slight slowing of flow in different endotheliums when they're when you need that kind of extravasation. So yeah, it wouldn't be that one. For this one, it would be E, because you get polymorphs infiltrating. So you get those like neutrophil types. So they're not mononuclear. So that's why it would be E. This one. So we definitely spoke about histamine. It's bound to heparin in granules as well. You also get tryptase, and you do get chondroitin sulfate. So it's a bit of a mean question because I didn't specifically say. So E would be the one that we don't find. But E is an important mediator that you do see, but you don't see it in granules. Okay, we're very nearly done. So just a quick note on chronic inflammation, because that will be kind of a good one to differ with. Differ with acute, and it could be kind of something that you might want to talk about in itself. So chronic inflammation, obviously going to be characterized by the time difference compared to acute. And it's also characterized by different cell types and by different mediators that are involved. So you could define it as kind of repeated bouts of acute inflammation, or it could be that you've got something persistently causing a constantly long inflammatory process, which would turn into this chronic inflammatory picture. When you've got kind of exhaustion of those initial mediators, and so then you're probably going to see a lot more of the chronic inflammatory mediators and chronic persistence of other different cell types, rather than the neutrophils that we're used to seeing acutely. So you're going to see more of the kind of lymphocyte picture as well, and macrophages, and less of the neutrophils. It could be that there's also excessive repair present as well. And a good example to learn is like a granuloma, because they're very specific. But the granuloma kind of concept will come up time and time again, especially if you do infection immunity, but it also comes up quite a lot clinically. So it's quite important to learn. If you, I would learn this definition of what it is, because it's really useful to know, and might come up in MCQs as well. And this is a good graph to have in your head as well, just to know that what ideally we want is we want it to be pro-inflammatory because it's the balance of clearing that infection, but we also then want it to be able to resolve afterwards. And that balance is really important. However, it's pathological when you get that persisted chronic or unresolved inflammation. And you get persistence of either an infection itself or various injuries and things like that. So this is an example of granuloma. So you can see the macrophages are here in the middle. These can be called, there's different types of macrophages, but they can be called like giant cells as well, you might have heard of, or like epithelioid macrophages because of their appearance and being very pink. So just think that really deep pink bit in the middle will be your macrophages. They will contain bits of pathogen, which eventually, once the granuloma is formed, those pathogens will die. But it's the fact that they weren't being able to be killed immediately with the innate immune response, such as with TB infection. And then round the edge, you have your lymphocyte cuff, so you'll get T cells and B cells outside of that. So that's an important thing to learn about. And in the middle, you might get that necrotic core. So it's called like caseous necrosis, which kind of means cheese-like. You might get that necrotic core in the center of your granuloma as well. There's a really nice table here, which I won't explain, but you can use that when you're revising of the different features. Quick question. So which of the following is not likely to cause chronic inflammation? Anyone have any thoughts on that? I think there's just 2 questions and we're done, really. So A, obviously we spoke about, is NADPH oxidase, mutation in that gene causing the CGD disease. So it's not going to be that one. Asbestos and silicosis we spoke about would be a cause. Yep, D is the correct answer. So exposure to a bee sting would be, you would get a certain kind of, you would get an innate initial response, but you'd also get a kind of a T cell mediated response with that one. So that would be D, and that would be acute. C, if we add an adjuvant to a vaccination, that kind of helps the innate immune response initially, and that can help, for example, in elderly patients when they get the flu vaccine, they will come with an adjuvant in them, and that will help kind of boost the local immune response, so they're more likely to form an antibody response to whatever you're injecting as well. And then, like I just said, with the granuloma, that would be a form of chronic inflammation associated with TB. Great. And then neutrophilia is caused by all of the following except which? Neutrophilia being increased levels of neutrophils. C, yeah, great. So viral infection is going to be causing a increase in the lymphoid compartment. So the B and T cell response is going to be a lot more important than your neutrophil response. That's essentially because viruses are going to be intracellular replicating pathogens, whereas the neutrophil can help destroy those kind of extracellular bacterial pathogens as well, and they're very good at that. Okay, so that's great. So if you would like a copy of the slides and to give some brief feedback, sorry, it was very, very quick going through. I tried to cover as much as possible. And yes, I'll put the link in the chat. That's fine. I'll stop sharing for a second. Did anyone have any questions they wanted to ask out loud at all? Or type any more in? I'll try my best to answer them. Or I can look them up. Feel free to email me and I can look them up too. And get back to you. So I hope your exam went well today as well. Someone told me you had exams. Okay, so I think that's the link. Let me double check. Yeah, great. So yeah, main take-home messages from that is kind of learn a few of the key mediators where they're really important and where they kind of have a key role where they go wrong, and just if there is a slight difference between different cell types, that might be something that they could quite nicely put in an MCQ. So it's kind of a lot of rote learning of different names and different receptors and things like that, but I'm sure you'll be fine. When do you have this exam? 20th. 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 20th Next week, Wednesday and Thursday. Okay, well, good luck. It'll be great. Hopefully there's some useful things on the slides, and like, yeah, make good summary tables can be quite helpful too. Thanks all for joining. Feel free to leave whenever you want. I'll just leave the thing on if people are still doing the feedback forms, but if not, thank you. Have a nice evening. Yeah, I think the link is in the chat. Oh, I think it might have sent just to one person. Sorry, let me try and send it again. Okay, I've sent it again. Sorry. It's all right. Thank you. That's okay. Thank you. Bye.
Bye, Molly. Thanks.
Bye.
See
Thank
you
you.
later.
That's all right. Hope you didn't have to listen to much of that.
No, it's great. Honestly, great job.
Oh, thanks.
I think
I feel like an hour is probably too much if it like any more than an hour would be too much because you're just trying to cover so much.
Yeah, absolutely. They did a great job.
I know. Have a nice evening.
Thanks, you too.